Oh, great. Hello and welcome, everyone. Thank you for joining us today. We're very excited to present this webinar today discussing new research results studying acral melanoma. You will hear from two outstanding MRA and CDMRP funded investigators. And for those of you that don't know me, my name is Dr. Marc Hurlbert, and I have the pleasure of serving as the CEO of the Melanoma Research Alliance. And today we also are going to be joined by MRA's Chief Science Officer, Dr. Joan Levy. She'll be helping moderate this MRA Meet Up session. This session is part of our ongoing Meetup series designed to educate, engage, and empower patients, caregivers, and the broader melanoma community. Today's Meetup focuses on acral melanoma, a rare subtype that can affect the palms of the hand, the soles of the feet, and the nail beds. And this rare form of melanoma remains a significant challenge for patients and clinicians alike. However, thanks to new data emerging from two of these investigators with us today and their global collaborators, we're beginning to better understand the unique biology, risk factors, and potentially treatment opportunities for acral melanoma. We would also like to thank everyone who helped make this event possible. Remind you that you can visit curemelanoma.org/blog and click the Patient Webinars tab to view our full library of past sessions. So just before we begin, I wanted to cover a few house rules. This is a public webinar and it is being recorded. The recordings will be available on MRA's website and may be shared on social media. This session is for educational purposes only, and it's not a substitute for professional medical advice. Please consult your healthcare provider for questions specific to your care. To ask a question, please submit it via the Q&A tab. And finally, please be respectful of the experiences, opinions, and expertise shared today. So it's my pleasure now to introduce our two panelists. First, we have Dr. Eugene Semenov, a board-certified dermatologist and co-director of the oncodermatology program at Massachusetts General Hospital and assistant professor in dermatology at Harvard Medical School. Dr. Semenov leads pioneering research into the epidemiology of melanoma with an emphasis on improving outcomes and treatment selection for patients with advanced disease. And next, we'll have Dr. Patricia Possik, International Fellow at the Sanger Institute and Group Leader at the Brazilian National Cancer Institute in Rio de Janeiro. Dr. Possik focuses on the cellular and molecular biology of tumors with a focus in her lab on acral melanoma. She recently published a landmark analysis examining acral tumor biology across diverse populations, and will be shedding some new light with us today. So thank you both, Dr. Semenov and Possik, for joining us. We'll get started moderating a panel discussion with both of you. I'll do parts of this. Dr. Levy will do other parts, and really look forward to get going. The first question to each of you, and we'll start with you, Dr. Semenov, is if you could describe sort of what has drawn your interest in acral melanoma, this rare and understudied subtype. And then Dr. Possik, the same question to you.
Wonderful. Thank you so much, Marc and Joan, for the opportunity to be here to spread the word about the work we do, and most importantly, to reach the patients and all other interesting parties who want to learn about acral melanoma. I'm a dermatologist, and of course, one of my career focus areas has been to try to improve the care for all melanoma patients. And though there's been tremendous influence of checkpoint inhibitors and novel systemic treatment options for cutaneous melanoma over the last 15 years, acral melanoma and other rarer melanoma subtypes have just not received as much attention. And their outcomes in these settings continue to be not as good, and in many cases quite poor, where the patients just don't respond to therapy. So understanding this gap and the very high unmet need, we were very interested in trying to form a better clinical registry, combine our efforts with many other institutions to get more access to acral melanomas so we can inform who gets them, why they get them, and also how can we better treat these patients.
Wonderful. And what's drawn your specific attention and focus, Dr. Possik?
So I'm a biologist in training, and I did my PhD and postdoc in the US and Europe. But when I decided to come back to Brazil, that's when I started focus on acral melanoma research. And that was because it was brought to my attention by clinicians and local scientists because of the higher proportion of cases that we have here, and maybe I'm in this big institute that has many cases and access to patients who want to participate in studies and contribute to studies. So as I became aware of this big gap in research, and my training was already in melanoma, I saw it as an opportunity to continue contributing to the field from Latin America.
Wonderful. Well, very passionate. So we're excited to hear about your results and some of the things you're seeing. Dr. Semenov, I was going to ask if you would add a little bit more about acral melanoma, sort of what it is, how it's different from other types. I mean, I think most of our viewers, when they think of melanoma, you think of a mole or a spot on your head or somewhere on your shoulders or body. And so we're talking about acral on the hands and feet. So can you explain it to us a little bit?
Oh, wonderful. So acral melanoma is traditionally defined by its location, as the name suggests. What we're in dermatology refer to as acral surfaces are going to be your hands, feet, and specifically, it's going to be the palmar hands, the plantar feet, and areas under the nail, so in the nail bed itself and nail matrix. These are traditionally under-surveilled areas because most patients don't pay as close attention to what's on their plantar feet as you could on a more obvious and easily accessible side of the body. And this is an area where the sun usually does not reach as often as it does on the trunk or on the face. So as a result, it doesn't really follow the classic adage of melanoma being caused by ultraviolet exposure or blistering sunburns. That unfortunately has also been part of the problem, because if the etiology, if the cause is different, then maybe the disease is different enough that it will not respond to the same treatment options. And that indeed is what we've been seeing. One of the issues that I alluded to and that really contributes to the poor outcomes here is that often patients go undiagnosed with these diseases because of the more difficult surveillance in order to pick them up. So they present or tend to present at more advanced stages where the disease is no longer necessarily just in the skin, but may have already spread to the lymph nodes or even potentially inside the person's body to other organs. And that also adds to the complexity of managing this population. The disease tends to be more on the feet than on the hands, but it can present in both locations. And what we have not necessarily appreciated as much, even though I mentioned it is largely on the palms and the soles and under the nail, it can actually also occur on the top of the hand, so on the dorsal surface, and on the top of the foot. This is part of the problem of studying acral melanoma in that we don't yet have the best definition. We know it occurs roughly in that general area. We know that there may be some similarities histologically. This is when we look at the melanoma under the microscope and try to understand what it looks like and is it similar or different from other melanoma subtypes. But there's still an ongoing conversation of what is an acral melanoma? Is it an entity defined by its histology? Is it an entity defined by the location where it arises? Or are there more specific molecular or genetic bases that can help us define this group together? So this is ongoing work. I think it's super important. It will allow us to better characterize the patient's disease and hopefully match the right patient for the right therapy.
Thank you. Thank you. Very, very helpful for us to better understand. And then Dr. Possik, first, I should have been much more enthusiastic. Thank you for joining us from Brazil. Very excited to have you here. But with that, would you share a little bit about geographic and demographic considerations, prevalence in certain populations in the U.S., Europe, or Latin America? If you could just give a little more insights there.
Sure. Well, thanks for the opportunity and for the invitation. It's great to be here and participate in this discussion. So as Dr. Semenov already said, it's difficult to diagnose a disease, especially because of all of the features that were already mentioned. But there is one thing, which is the proportion of acral melanoma cases across the different types of melanoma, include those related to sun exposure. So those related to sun exposure, those that we are more familiar with, they are associated or more common in people with fair skin, a history of sun exposure or burns, a larger number of moles. And these are populations usually from Europe, from the US, from European descent. And then in other populations, like in Latin America, in Asia, in Africa, these cases are not that are rare compared to Europe and the US and Australia because of the type of skin and other features. So the overall number of cases of acral melanoma cases, the relative number of acral melanoma cases compared to the other subtypes tend to be higher but that's because the sun exposure related one is lower. So it's not the acral melanoma is more common in people of non-European origin. It's just that proportionally we have more cases. And in countries like Brazil, Mexico, or China, and some countries in Africa, and countries of non-European or admixed populations, you tend to have more cases of this type of melanoma compared to European descent populations. So the overall incidence across populations is similar, regardless of skin color ancestry. This is important to know. And so I think that's why we see so many more cases here, and the importance of this tumor in these countries is a bit more taken into consideration when you're doing research and clinical care.
Thank you. We won't make the audience listen to our biostatistics analysis, Dr. Semenov, about these incident rates across populations, but it's more common among people with darker skin tones than it is in more Caucasian skin tones. Proportionally, then, it's much more common in Brazil, as you've seen, and you'll share a little bit more Latin America and Asian countries. So I think it's very helpful sort of grounding the understanding for everyone to know what we're talking about. So I think we will move on a little bit, Dr. Semenov, to really a groundbreaking project that you and myself and Dr. Rebecca Hartman and some of our strong colleagues from other institutions have led, and very exciting. We were assembling the largest cohort of acral melanoma cases of patients with melanoma, and looking at what are risk factors for these rare subtypes, because we don't think that it's always related to sun exposure, or in fact, we know it's not related to sun exposure. So I'll dive into Dr. Semenov, if you could briefly explain some of the clinical environmental risk factors that have been identified.
Yeah, so thanks again. I think this is a very important question. And to really understand and gain an appreciation for the paucity of data here for risk factors, it's often helpful to review what are the risk factors for cutaneous melanoma, where we've had a ton more work and a lot more research done. There, of course, and I'm going to go through this rather quickly because a lot of this is probably familiar, the most common risk factor that we think about is ultraviolet exposure. And the populations that are at highest risk for developing DNA alterations to the UV exposure are going to be more Northern European descent Caucasian individuals, where the common cutaneous melanoma is seen almost predominantly. Male sex has been associated with increased risk for cutaneous melanoma, all age over 50 years, and various germline mutations. For example, there have been multiple familial mutations that predispose to melanoma in the individual's genome, like CDKN2A, that we'll talk about shortly. Patients who have had organ transplants and other reasons for immunosuppression, so where they're taking medications that reduce their body's ability to fight off infections, have increased risk of melanoma. Ultraviolet exposure that also can be manifested by using tanning beds or indoor tanning beds was a very, very high risk. And in people with many, many moles, some of which may be atypical, or what we call dysplastic nevus, or just a sheer number of moles. As that number increases, there tends to be a greater risk that one of them may start misbehaving and potentially eventually become a melanoma. So that's what we know in a very quick nutshell about cutaneous melanoma. In acral melanoma, these types of studies have been very challenging to do, in large part because the melanoma is rare. About 4 to 5% of all melanoma cases, at least in the US, are acral melanomas. And one of the challenges that we face here is that our healthcare systems do not always talk to one another. For instance, I may see a number of acral melanomas here at Mass General, and someone at MD Anderson may see another couple. But what we don't do historically, because there isn't a great central source outside of the SEER, which is a very large cancer registry across the US, but there isn't a way for us to combine these easily in order to study them at a population level and study them not just from an individual perspective, but also molecularly. So that's always been an impediment in the US for us to gather our numbers together and find a way to really understand the etiology of this disease. So the project that you're leading through a grant by the Department of Defense was really one of the first comprehensive efforts to combine a number of groups across the country and collaborate with individuals even in other countries, at least to gather the data that has already existed in order to understand what are the risk factors for these melanomas. Historically, what people have suggested as may potentially be implicated in acral melanoma pathogenesis are trauma to the area. So individuals who have, like athletes, for example, who have repetitive trauma to their feet, there's been some concern that maybe they're at increased risk of melanoma. Individuals who work outdoors with heavy machinery, so construction workers who may have a lot of repetitive vibration-induced injuries to the hands, are potentially at higher risk of acral melanoma. There's been some possible linkage between increased BMI or obesity, as it may also pertain to some additional stress and pressure on the feet, for instance, and that could possibly predisposed to acral melanoma. But overall, these risk factors have been very poorly validated and extremely poorly understood. So it's unfortunate to say that the field, as it has existed before the start of our study, has had very little understanding of what the risk factors are. If I may, and I'm happy to go into some of the risk factors we did identify, Marc, through the work that we've done. So in collaboration with investigators at the Veterans Affairs Administration, at the Brigham and Women's Hospital, at Colorado University, and across multiple other sites in the US and even abroad, we've come up with a fairly large cohort of acral melanomas, approximately 2,000 patients, which again is an extremely comprehensive population that outpaces by a factor of magnitude all prior studies. And what we've seen there, interestingly, was that Agent Orange exposure, and this was a study led by Dr. Rebecca Hartman from the Veterans Affairs Administration, actually increases the risk for acral melanoma development by more than 30%. There were other risk factors that we identified, and this dovetails off of was just mentioned about different racial predispositions. For instance, even though we know that cutaneous melanoma is primarily seen in Caucasian populations, and acral melanoma can be seen in a population representative manner across all racial and ethnic subgroups. In this study, we actually saw that there was, even despite that and beyond that, an increased risk among the Black population as well. In fact, there was over a 60% risk in the Black population by comparison to the reference group being Caucasian of developing acral melanoma by comparison to cutaneous melanoma, and this is across their lifetime. And even by comparison to a general population, the Black population had a much higher risk of developing acral melanoma in their life. So what that suggests is that, yes, we commonly thought of this disease just being agnostic of where the individuals come from, what their racial backgrounds were. But there may be some additional susceptibility amongst some racial groups that we need to study more carefully. Interestingly, one of the things we found that we're still trying to figure out is that current smoker status was protective of the development of acral melanoma. And this is, not a license to go out there and start smoking, but something that actually has been seen in a couple of other cancers where there may have been some protective effect as well. And that still remains to be elucidated why and how. Maybe it's a competing cause for something else. Maybe these patients are not living as long because they're developing other diseases and therefore don't have the chance to develop acral melanoma. It's not entirely clear, but we did find several interesting risk factors here. We're still trying to analyze this data, so these are findings that are not complete, although several of our early studies are about to be published, including this one that's led by Dr. Hartman. I'm happy to go into further detail, Marc, if there are additional follow-up questions here.
No, I think this is very helpful. And again, if anyone in the audience wants to ask a question, please enter in the Q&A and Dr. Levy will get to that. I think if you could just comment, one of the things I hear from the patients that reach out to the MRA is, I don't understand why I got this, acral melanoma on the bottom of my foot. Could I have gotten, inherited some bad genes from my mom and dad? Is there something that could be a genetic risk factor? Where are we at in identifying those? Most people have heard of BRCA for breast cancer. Where are we at for acral melanoma?
Absolutely, another very, very important question. I think we all would love to be able to find out very early in life if we're at risk for developing different conditions, be they cancers or other conditions. And indeed, there's a whole line of research that aims to study this by investigating the individual's genome. So we look at what we call germline susceptibility or germline risk factors. In the classic cutaneous melanoma setting, this has been done more elaborately. For instance, one of the common familial mutations that people identify as increasing the risk for developing cutaneous melanoma is in the CDKN2A gene. It's a cyclin D kinase gene. There are others, in CDK4, for instance, POT1 is another one. But the field is far more evolved there because there are so many more patients to study. In the acral melanoma setting, an individual gene has not yet been identified because most of the time the acral melanoma cases have been combined with the cutaneous melanoma cases in larger studies. And subgroup analyses of acral melanoma specifically have not been powered sufficiently to be able to do an independent analysis. So some people would argue that maybe there are shared susceptibilities, maybe there is going to be a CDKN2A component, because again, in the overall large group analysis, it came out to be the case both within the broad melanoma population inclusive of acrals, but not individually within the acral group exclusively. So what we're doing currently as part of this Department of Defense funded project is we have collated the germline data across multiple institutions in order to really investigate this question thoroughly. We will have in this particular cohort over 800 patients that we've been able to get through public sources. So these are things that have already, studies that have been done that have happened to germline sequence patients who have had acral melanoma, as well as our own cohorts that we've undergone sequencing as part of the project. So we're combining all of these various resources to try to understand, are there specific mutations that are inherited by the individual from their parents, or potentially even new mutations that a person develops in utero, and could those be responsible for why they later on in life develop acral melanoma? As with everything in genetics, and germline genetics specifically, it's often difficult to assign all the blame just to one gene. These tend to be polygenic in nature most of the time and often require multiple hits. So you have the predisposition, but not everyone with the predisposition may develop the disease. So there may be something else that happened later in life, like trauma, for instance, to the area that could have been that second hit. So it still remains to be seen. I do think that there will be some shared susceptibilities between acral melanoma and cutaneous melanoma, as is evidenced by the fact that people who get melanoma can get both. And in fact, there's a slight increased risk for people to get these rare melanomas if they already have cutaneous melanoma. But it will probably not explain the whole story. And that's why we need to do the study, because patients deserve to know, are they at risk? If we can even offer some information in that regard, I think it will be helpful, because like you said, in BRCA screening, we would be able to counsel patients that they may need to undergo mastectomies prophylactically to prevent the development of cancer. But with melanoma so far, especially acral melanoma, we don't have that level of certainty. And this is why we're doing the work we're doing.
Well, thank you for that. And thank you for your hard work on this project. To put it in context, some of the most seminal papers on acral melanoma studied 30 patients or 50 patients, and now you've assembled a cohort of 2,000. So really wonderful work. So I'm going to hand it off to Dr. Joan Levy in a minute, but just to put it into context for all the listeners, so we've now been focusing the first half on what are the risk factors for getting the disease of acral melanoma. Now we're going to shift gears to look at the biology of the tumor once you have acral melanoma. So moving from risk to tumor and tumor biology. So Dr. Levy and Possik.
It's a pleasure. And I just want to thank my colleague, Dr. Marc Hurlbert, for leading this CDMRP melanoma research project, and Dr. Semenov and his collaborators for coming up with and analyzing such important risk factors and helping to identify them, and now moving on to germline and genetic factors. So Dr. Possik, it's a pleasure. I just want to start off with saying MRA has supported a number of research projects to better understand tumor biology and perform molecular analyses on all subtypes of melanoma, including acral melanoma. Primarily, they've been from Caucasian populations of European descent. We talked a little bit about this before with respect to the epidemiology and incidence, but your landmark study really helps analyze acral tumor biology across diverse populations in a Brazilian population. So could you start off telling us more about the populations you studied and some of the recent tumor analyses in those populations?
Sure. So first, I want to give one comment about congratulate Dr. Semenov, because it's an example of how investing in studying rare diseases can change the way we think of the disease. So it's great that you have such a cohort, and I'm looking forward to see the results in practice. So here in Brazil, as I said, we have access to many patients who want to participate in studies, both clinical studies, but also experimental studies, by contributing with data, with information about their disease and their care, and also with samples, biological samples. So these patients, they are treated here at the Brazilian National Cancer Institute. We invite them to participate in our study. And once, if they agree, we get, during surgery, we get a piece of their tumors and preserve it alive so that we can bring it to the lab and establish experimental models. Meaning, so we cells that we will put in culture and they will live forever. Or we can inoculate in a mouse, and the cells will be live in the mouse and also live forever if we keep them growing or giving them food. So we do that in parallel to characterizing the genetics of the tumors and other analysis of patient clinical data, histological data, and combining with these models. So we did this effort together with different collaborators in different countries, including in Mexico, Dr. Daniela Robles Espinosa, who's doing similar work in the Mexican population, and also US and in the UK, that made this possible. And this also got funding from the MRA and other foundations that help this, because the situation of funding in these countries, Latin America and Africa, is not locally enough to conduct such a work. So I'm very glad of this international funding. So once we establish these models, we can, and we know the genetics of the tumor, so we know, as Dr. Semenov was saying, like which genes are mutated, we can manipulate it in the lab and understand if the cells are dependent on this gene. So for example, we can turn on and off a gene that we identify as being mutated in acral melanoma and see if the cell will survive or if the cell will die, either in vitro, in a cell culture, in a plate, in a dish, or inside a mouse, subcutaneously. We can also understand if these cells are able to metastasize to different organs in the mouse. And we can follow that and understand, or at least ask or investigate which genes or which factors are involved in this metastatic dissemination. So that's what we did here. And we did all of these experiments with different strategies. And now we are trying to understand what are the low-hanging fruits, like the genes that are potential targets that can be further studied in preclinical and maybe clinical settings. And we're also now have this collection that we are soon gonna share it through the MRA catalog of models so that more researchers can access these models and ask their own questions, because as we are discussing here, there are so many questions to be answered, and that's the idea of these collaborations.
Yeah, so I just want to point out, it's phenomenal, global collaborations, just are sometimes very difficult to do, but you've done a great job of being able to involve many different countries and many different really well-established investigators in this analysis. So could you just give it a very simple and high level, like what are some of these alterations in the the tumor samples that you have performed analysis? And are there differences between acral melanoma and cutaneous melanoma per se?
Yes, we saw several differences when compared to cutaneous melanoma. Like, for example, one of the most common mutations in cutaneous melanoma is BRAF, which we have target therapy to tumors that are specific for BRAF mutations. So in our cohort, we saw less than 10%, while in cutaneous melanoma, this number can be over 50%. So in terms of driver mutations, that's, I think, the biggest difference. But in terms of other type of alterations, which is when you have a gene that is not mutated, but you have several copies of that gene duplicated in your genome, and that makes that gene more potent, can make this gene more potent. So that type of alterations, we saw many, which is not common in cutaneous melanoma, and they involve genes that were already identified as relevant in acral melanoma, such as CRACL, CDK4, TERT, telomerase. So these are known candidates that we were expecting to see. And by saying that, I think you can already appreciate that we saw the same thing, or at least we think we did. We don't have a very different result from what we see if we analyze an European acral melanoma cohort or an Australian acral melanoma cohort. So the mutations and genetic alterations are quite similar if you compare across acral melanoma studies.
Yeah, so just one last point. How can these insights inform new therapeutic strategies? Like you're identifying different alterations, and you've mentioned that there's a drug against BRAF mutations. Has this provided alternative therapeutic strategies? Because we know that acral melanoma is not as responsive to the same therapies as cutaneous. So if you could just comment on that, that would be great.
Sure, yeah, because BRAF mutations are much less common, a lower percentage of patients would benefit even to have access to this therapy. So 90% of the patients, we would have to think of alternative treatments. And so by doing this type of analysis, you can identify sometimes even like genes that we already know, drugs that are specific for that specific protein, like CDK4 inhibitors or even MDM2 inhibitors and other inhibitors that now we are exploring by performing large-scale drug screens in our models. So we are testing different drugs, different pharmacological agents in our cells to see how it relates to their genetic status. And also, so this is how we can get this information. And I didn't tell you much about the immunological angle of our work, but we can also explore these differences, the differences in the immune infiltrate and how the immune cells recognize and fight tumor. And understanding this difference will also help us understand which immune therapy is more efficient for acral melanoma, or if it's similar to the benefit that cutaneous melanoma patients will have. So this is the type of information we can take from these studies.
Yeah, that's a phenomenal amount of information. A big congrats on this large analysis, not only providing information on different molecular alterations, but potential new therapies and the way to involve the immune system. That's really phenomenal work. And also, Dr. Possik said before that they're going to be able to share these precious models, globally. And that is really important so that future acral melanoma research can be conducted by other labs. So at this point, I just want to comment and say many times patients and caregivers ask, how can I get involved in research? And just wanted to make those on the webinar today aware of a project that the MRA is sponsoring, which is called the RARE Registry. And it's a direct-to-patient registry for acral, mucosal, and other rare melanoma subtype and cutaneous melanoma patients to participate in. There's a series of surveys they fill out, and it really helps researchers and clinicians understand the patient journey of acral, mucosal, and cutaneous patients. And you could find more information about this registry at raremelanoma.org. I also just want to also mention that we are having a patient forum on February 25th in Washington, D.C., and you'll be able to sign up for that. If you can't attend in person, it is held live, and the communications team creates enduring material. It's a great way to meet other patients, caregivers, and hear phenomenal talks from investigators. So at this point, I just want to ask each speaker to give just a 1-minute closing remark on the remaining gaps and future priorities that would be important to drive acral melanoma research forward. Dr. Possik, first.
Oh, I just want to thank everybody who's interested here and listening, because I think you already recognize the importance of studying acral melanoma or any disease that is rare, but still is a burden to patients, their family, and entire populations. But I hope we have convinced you that acral melanoma studies about rare subtypes like acral melanoma is not justified, but it's essential and for all populations. And studies, larger studies in diverse populations, including like large studies like Dr. Semenov mentioned, are crucial for us to get this information that will translate to better diagnosis, better treatment, and better outcomes. So I think we are on the right way, and I thank a lot MRA for the support, because you're making a difference.
Thank you so much, Dr. Semenov.
Yeah, so again, thank you for raising awareness about this very important topic and a very understudied disease. One thing that I would like to highlight is that this research can only be done by patients who come forward and provide us tissue, provide their experience about the living with acral melanoma, so we can understand maybe why they have developed the disease, and more importantly, understand how we can support them better as they go through their treatment process. Thinking about current gaps, in addition to what we mentioned before, we do know that immune checkpoint blockade has been a godsend, essentially, in managing melanoma in general. It's revolutionized our care of all melanoma, but in the acral setting, the response rates are still below 1/3 of the patients that have objective response to checkpoint blockade. So some of the novel strategies that are currently on the horizon and we think are going to help, and certainly the early data seems very hopeful, are going to be intralesional therapies. So if only 1/3 or less of acral melanoma patients are responding to checkpoint blockade, it must mean that the tumors are either what we call cold and don't have a lot of immune infiltration, and therefore not much of a fertile bed for the immunotherapy to work on, or there's something else that's inhibiting the immune response, even if the immune infiltrate is present. So one of the mechanisms that we've tried as a community to transition a cold tumor into a more hot tumor have been these intralesional therapies, like TVEC has been introduced a number of years ago. And more recently, there is a new trial called IGNITE-3. It's a phase 3 trial where they're combining another viral vector. So it's a herpes virus that incorporates a GM-CSF, granulocyte monocyte colony stimulating factor. And what that does is it helps recruit immune cells into the area where this factor is injected, and that the virus essentially enters cells and helps replicate more of this factor to attract more immune cells. So it's been very reassuring. In this particular trial, where about 18 acral melanoma patients have been enrolled, the response rate actually has been about 44%. So already, and this is again patients who have already failed immunotherapy as a first line. So this is a population that hasn't done well to begin with. So for the response rate to be higher than in the first line setting is quite encouraging. We also have done quite a bit of work in acral melanomas, and specifically in transit metastasis from acral melanomas using TVEC, which is the predecessor medication to this new drug that's under investigation. And even there, we've had some hopeful responses where we can control these tumors from further spreading into other organs. So we can try to keep them at bay while they're in the skin. So I think that the opportunities now are try to get earlier diagnosis through better screening, and that will involve a better understanding of risk factors and maybe more concerted screening efforts in the high-risk populations. And then separately, being more aggressive in managing disease from an earlier stage, because this is a disease that tends to spread rather quickly. It's more than twofold more likely to have metastatic spread than cutaneous melanoma. So there must be a way that it evades the conventional surgical treatments. And I think that what will bridge that gap is being potentially more aggressive with these intralesional therapies that are hopefully going to be far less toxic than the systemic immunotherapy. So there could be a bridge. Treat them early, treat them more aggressively, surveil them more closely, and hopefully we'll catch them before they spread. So I think that will be the new paradigm, and I'm very hopeful that we'll be able to get there altogether.
Yeah, well, thank you so much for that information. And you mentioned the importance of patients donating samples and tissues and all, and I'm excited to share that the MRA just opened an MRA Melanoma biorepository. It's being housed at the University of Colorado, and any melanoma patient with any subtype can contribute tissue and tumor tissue and normal samples. So please go on to our website and find out more information. I just want to have, for the remaining few minutes, just to ask a few questions. And to Drs. Possik and Semenov, and one of the questions is, do you see a different rate in metastasis or differences in metastasis, between different patients with acral melanoma of different races or ethnicities? Dr. Possik, do you want to comment?
I can comment, but I'm sure Dr. Semenov can continue from where I live. But I don't think there is enough evidence for that. But as we were discussing here, we still have so many studies to reach a consensus. And what if acral melanoma is more or less aggressive than cutaneous melanoma? That's still too early to really understand if there is no difference. But I think we need to consider other differences that make a difference in patient care, like the Dr. Semenov was mentioned. Sometimes or most times they are diagnosed late, so it's not related to biology. But when these patients come to the clinic, it is already metastasized or close to metastasize. Awareness, so raising awareness so that people can come earlier and check, a suspected lesion. I think these can make a difference, but not related to biology. And access, like maybe regions that have or countries that have less access to dermatology clinics or to medical doctors that can diagnose or offer any treatment early enough. So I think that's all I can contribute. Dr. Semenov, if you can take.
Well, that's a very large contribution. I echo everything you've said. The work that we have done that actually was somewhat hopeful is that after you adjust for stage at presentation, so with the caveat, of course, that some populations present at later stages, especially populations that are not living close to NCI-designated cancer centers or places that you have large expertise in melanoma and specifically rare melanoma. But socioeconomic factors absolutely play a role in that. But if you adjust for stage at presentation, there really isn't any difference in metastasis rate between both acral and cutaneous melanoma, and we have not yet seen well, so that's not true. So there's no difference in the rate across races. in those two settings. There is a difference in metastasis rate between acral and cutaneous melanoma because acral melanoma tends to metastasize at twofold, so twice as likely as cutaneous melanoma. But racial differences we have not seen. Where the racial differences come in is largely differences by proximity to center. So unfortunately, in this country, I think there are multiple underrepresented groups in healthcare that don't have the same access to medicine, and they tend to present at later stages to begin with. And when that happens, it's just that much easier for the disease to then become metastatic. I think that will also play a role with access to systemic therapies. If a person doesn't have the best insurance, they may be kept on therapy, at least initially, that maybe isn't as great up until they can get the right access to the more combination immunotherapy or other approaches that are maybe more clinical trial in nature, and they need to be in particular sites to be able to enroll in those trials. So I think that that's a non-biological source of disparities that we absolutely need to address better. But all of it starts with education and being able to disseminate what we do know everywhere. We need to educate people how to look at their own skin in a way that can capture these as early as possible and then be able to get to the right places for management. So if we can solve that, I think we're going to be able to reduce some of the disparity that we're seeing.
Yeah, and maybe just a comment on Dr. Semenov on the importance of clinical trials. I mean, as you know, everyone's heard today, the standard of care for cutaneous melanoma doesn't seem to be as effective for acral melanoma. So, you know, what about clinical trials? Dr. Semenov mentioned one, but there are other clinical trials that acral melanoma patients can participate in, and they could also find information on these clinical trials on the MRA website. Would you like to this is the final question, Dr. Semenov, comment about it?
For sure. I think that this space is going to be growing quite rapidly in general because finally we have a growing body of scientists, clinicians who are able to collaborate together with the MRA and organizations that are very interested in supporting this work to be able to fund the right studies. So some of these studies are, of course, in the pilot setting being funded by at the behest of the investigator using foundational sources and federal sources like MRA, NIH, Department of Defense, to try to understand, can this treatment work better in acral melanoma than other things that have been done before? And for that, patient participation is absolutely essential because we learn everything by access to the right tumor tissue and the patients who are willing to participate. And then ultimately, maybe repurposing the medications we have available to us already, but in more aggressive regimens. So instead of doing single agent monotherapy, which we don't typically do anymore for more aggressive disease, and going to dual agent immunotherapy, so not monotherapy for immunotherapy, but dual agent, is probably going to be the new standard of care for a lot of these patients of acral melanoma. And that may be preceded by some of these intralesionals, like I mentioned. So you prime the tumor, hope to get some more fertile soil for an immunologic response, and then do immunotherapy with the intent that maybe at that point, the tumor will respond better than it would have otherwise responded if you just challenged them with immunotherapy to begin with. This is also some of the questions here will be the adjuvant, neoadjuvant debate that permeated a lot of the melanoma space. So this is, I promise, one final minute here, but historically we've treated advanced tumors by first cutting out the tumor or as much of it as we could, and then giving immunotherapy. But now we're favoring more giving the immunotherapy or other systemic therapy first, and then getting a response, and then cutting out whatever tumor remains, and sometimes following up with more immunotherapy in the adjuvant setting. And that has been shown to lead to better recurrence-free survival. So it is my hope that we can do similar studies focused on the acral melanoma space, where again, getting large consortia and collaborations are going to be essential to get the sufficient number of patients to demonstrate benefit. Because we can't just assume that because it works in cutaneous melanoma, it will work in acral melanoma. So it has to be directed to the subtype to some extent.
Yeah, well, thank you so much for this incredible discussion. Dr. Possik and Dr. Semenov, thank you for sharing your expertise, taking the time out of your busy days, and really helping enlighten those on the webinar of the important work that you are leading, and to better understand and treat acral melanoma. I also want a heartfelt thank you to all the patients, caregivers, and advocates joining us today. Your engagement and curiosity really helps drive progress and really inspires the MRA mission. And finally, thank you to everyone at the MRA who helped make today's MRA meetup possible. These meetups are, again, enduring materials, so they are on our website, and the recording of this webinar will be available in the upcoming days. So thank you again to all, and enjoy the rest of your day.
Thank
Thank
you
you,
for having us.
everyone.
Thank you.